Hi all. So I'm going to talk about the physiology. Hi all. So I'm going to talk about the topic physiology of excretion. Excretion in insect is a fundamental physiological process vital for maintaining homeostasis. The excretion mainly eliminates the metabolic waste, regulates osmotic balance, and conserves the water. Insects inhabit incredibly diverse ecological niches from freshwater to deserts, and their excretory physiology is highly adapted to these environments. The system primarily involves Malpighian tubules, hindgut structures, nitrogen waste pathways, and complex neuroendocrine regulation, forming an integrated mechanism of waste removal and water conservation. The integrated excretory system. The insect excretory system primarily involves Malpighian tubules and hindgut structures. These components, along with nitrogen waste pathways and complex neuroendocrine regulation, forms an integrated mechanism for waste removal and water conservation. So coming to Malpighian tubules, its major function is the urine formation. The primary urine formation is taking place in the Malpighian tubules. The hindgut structures. Hindgut structure is responsible for urine modification and water reabsorption. The nitrogen waste pathways is responsible for the diverse waste elimination strategies. And lastly, the neuroendocrine regulation tightly control over the excretory processes. Malpighian tubules, the primary filter. So Malpighian tubule is known as primary filter and they are long, slender, blind ended tubular structures extending freely into the hemolymph. Their number varies widely from a few in hemiptera to over 100 in locust, originating at the junction the Malpighian, the midgut and hindgut. So coming to the structure of Malpighian tubules, lined by a single layer of epithelial cells with dense microvilli. The cell membranes contain ion pumps, co-transporters and aquaporins. Malpighian tubules are rich in mitochondria for active transport processes. Their major functions are the primary urine formation by secreting ions and nitrogenous waste directly into the tubule lumen. Unlike vertebrates, insects do not use filtration. Instead, the excretion is predominantly secretory. These are the main excretory organs in insects. These are fine, unbranched and thread-like processes. These are the outgrowth of alimentary canal and present at the junction of midgut and hindgut. The Malpighian tubules are absent in springtail and aphids. They vary in different insects. In scale insects, there are 2 and 4 in bugs. In mosquitoes, the Malpighian tubules are 3 and 6 in moths and butterflies. 60 in cockroach and more than 200 Malpighian tubules are seen in locust. The walls of Malpighian tubules consist of 4 to 6 large epithelial cells arranged in a circle which externally rest on the basement membrane. It divides into 2 parts. The distal secretory part having honeycomb border, and the second part is proximal absorptive part having brush border. Coming to the mechanism of urine formation from dilute to concentrated. The urine formation begins in the Malpighian tubules and it is modified in the hindgut, a crucial adaptation for water conservation. The urine modification in hindgut. The hindgut consists of the ileum and the rectum, both of which play roles in selective reabsorption. The ileum, which absorbs the ions such as sodium, potassium and calcium, begins the process of water conservation. Coming to rectum, it contains specialized structures called rectal pads or papillae. It is extremely efficient in water and electrolyte reabsorption. This occurs in the Malpighian tubules. The active transport of sodium and Na+ into the lumen is driven by proton pumps. Chloride ions follow passively maintaining ionic balance. The water enters osmotically. Uric acid and other nitrogenous waste such as xanthin guanine precipitate as crystals due to low solubility. This results in a dilute primary urine which passes into the hindgut. Nitrogen excretion pathways. Insects exhibit versatility in nitrogen excretion, adapting their pathways based on habitat and life stage to optimize water conservation and toxicity management. Eurycotylism. The first one is eurycotylism. The most terrestrial insects excrete non-toxic insoluble uric acid, minimizing water loss, ideal for arid environment. Ammonotylism. Aquatic insects diffuse ammonia directly, requiring much water but less energy. Ureotylism. Some insects excrete urea alongside uric acid, particularly in moisture-rich habitats or specific developmental stages. Storage excretion. It's unique to insects. The uric acid can be temporarily stored in fat body cells, especially during metamorphosis or diapause. The osmoregulation and ion balance. It maintains the equilibrium. Insects must maintain ionic equilibrium despite their small size and large surface area. Both Malpighian tubules and the hindgut play critical roles in the delicate balance. The Malpighian tubules. They actively regulate hemolymph ionic concentration, adjust sodium and potassium secretion based on diet and habitat. They also remove toxic ions and metabolites. Hindgut. The rectal pads regulate sodium, calcium and potassium reabsorption, maintains hemolymph osmolarity, and they also helps adaptation to saline or drought-prone environments. Hormonal and neural regulation. Insect excretion is tightly controlled by sophisticated neuroendocrine mechanisms, ensuring rapid and precise adjustments to physiological needs. DH, the diuretic hormone, it stimulates Malpighian tubule activity, increasing ion secretion and urine flow. The antidiuretic hormone, ADH, enhances water reabsorption in the hindgut, crucial during dehydration. Neural control. Neurosecretory cells modulate tubule function, enabling rapid hormonal adjustments. Ecological adaptation. The insect exhibit highly specialized excretory adaptations shaped by their diverse habitats, allowing them to thrive in challenging environments. There are desert insects, blood-feeding insects and plant-feeding insects. So coming to desert insects, the efficient rectal pads produce almost dry excreta which minimize the water loss. Blood-feeding insects. The rapid diuresis after blood meals eliminates excess fluids, preventing flight impairment. Plant-feeding insects. They produce honeydew to remove the excess water from high sugar intake. Excretion is not merely about waste removal. It contributes to numerous physiological functions essential for insect survival and overall homeostasis. The waste removal. It eliminates the nitrogenous and toxic waste. Second, pH and ionic balance. Maintains hemolymph pH and ionic equilibrium. The water conservation. The water conservation is crucial for terrestrial environment. The metabolic support. Metabolic supports metabolic activities during metamorphosis. Detoxification. It facilitates detoxification of plant secondary metabolites. The physiology of nitrogenous excretion in insects, differentiating between terrestrial and aquatic insects. The terrestrial insects excrete uric acid or urates, which are water insoluble and require minimal water of removal. The aquatic insects excrete ammonia, which is water soluble and requires more water for removal. The process involves the secretion of ions into the blood by the Malpighian tubules, secretory region to form urates. The other excretory organs. The nephrocytes. Nephrocytes are the cells that save the hemolymph for products that they metabolize, pericardial cells. Integument. They are the outer covering of the living tissues of an insect. So next is tracheal system. Tracheal system consists of tracheae and tracheoles. Fat bodies. A loose or compact aggregation of cells, mostly trophocytes, suspended in the hemocoel, responsible for the storage and excretion. Rectum. The posterior part of the hindgut. Enocytes. These are the specialized cells of hemocoel, epidermis or fat body with many functions, and the major function is the excretion. Thank you.